So actually, Brennan, the first thing I'm going to ask, do you mind coming up here for this one? We're going to be doing some pairs really quickly. So today we're going to be talking a little bit about, this will be the first lesson in a unit on DNA to protein. So talking about some transcription, some translation, things along that whole route. So the first thing I'm going to do is something called a take 6. So if you guys don't mind passing these out to you, we'll go ahead and flip to the back side. And you guys will not need any of your own paper or anything for today. Two of you. And then each group is going to need a die. And so the very first thing we're going to do before I tell you anything is we're going to do, has anyone heard of Take 6 before? Yeah. Cool. So basically the way it works is you're in pairs and one of you is going to be the roller and one of you is going to be the writer. So we're going to be on the back side right now. So we've got this guy right here, the top, the Take 6. And we're going to take just a few minutes, kind of see how everyone's doing. But one person is going to start by rolling, and they're just going to keep rolling the dice until they roll a 6. And while they're doing that, the other partner is going to be writing anything you can think of that has to do with the topic DNA to protein. So literally anything that you think is relevant. So anything like that. And then once you roll a 6, you switch roles, and then you kind of keep going. And we'll just, I don't want to give too set of a time limit, just kind of see how everyone's doing. So whenever you're ready, you can go ahead and start.
Right. Just kidding. No. 2, 6. It's like weighted on that side.
Give me 5 to watch.
I'm just going to hang on to these for later.
That was 6.
All right.
No, that doesn't make sense.
I couldn't get it.
That's totally fine.
There's like, I think, you're wrong, isn't it?
I'm happy these died worse. All right.
Oops.
We'll go for like 2 more minutes.
Steal it, are you? Are you? Huh? So you're gonna steal it, are you?
No, I'm actually putting well thought out intellectual things. Just putting everything to the front.
No.
I have some extras, so if either of you would like. This is that.
DNA to protein.
Sam, you can just kind of join up with one of those groups.
Thanks.
This is for you. This is well. Thank you.
No.
Sam, why don't you jump it over there?
Okay.
Oh my gosh. Okay. That was
Yes.
That's not kind of good.
You need to whatever comes right.
Okay.
And like
20 more
keep
seconds.
writing it until she writes it. I wasn't trying to confuse her.
Go ahead and make this your last roll, or last turn.
A lot of 3s in there.
There's
We all good? Cool. So, just kind of to gather all of our thoughts, and anybody can jump in here whenever you want. What are some things that you had for DNA to protein? Any that you think are like particularly relevant? Kind of just get like an exhaustive list almost. Yeah.
Amino acids.
Amino acids. Cool. Anybody else? For sure. Yeah, feel free to just throw them at me. RNA.
Anticodon.
Transcription, translation.
Okie doke. Yep.
mRNA. Ribosome.
Alright, transcriptions and alright, RNA. You said what kinds of RNA?
mRNA.
Yep.
Also tRNA.
There's one more. Yep. There's more than one more. Anybody else? Ribosomes.
It's a protein because I like the protein thing.
Yep. Anything else? Anybody had? Alright, cool. So this is the first class of what would be the larger unit of DNA to protein. So at some point throughout this, we are going to be talking about all of these for sure. But today's focus is specifically transcription, kind of getting you guys introduced to these ideas. So codons and anticodons. This can be generally a pretty confusing thing for a lot of people to grasp. It's a little weird to think about. So this is going to be the main focus for today. Eventually we'll see how we start with DNA, and then through these we'll get to proteins and amino acids and so on. So if you can go over and flip to the front, we're going to work on the RNA transcription adjunct display. So in this case, an adjunct display, those can be pretty much anything, honestly. So we're going to be making pictures or kind of posters that ideally we'll put up along the walls. Obviously we can't do that in this context, but that's what we would be doing. And so we're going to be doing that by coding our own names. So I'll go ahead and read the directions aloud. But using anticodons corresponding to each letter, find the codon sequence corresponding to your first and last name in the work area below. Then write your name's DNA codon sequence on your blank sheet of paper and take some time to personalize it and design it. When you receive the paper of another student, transcribe their name onto the paper, going from codon to anticodon to the letter. Note any mistakes you may find in your partner's code on the back of their paper. So basically what we're going to be doing here is you have every letter of the alphabet with a corresponding anticodon. So for me, here, I'll do this up here. I'll write out Benjy. And then I'd personally recommend just doing it like this, and then writing the anticodon sequence here. Then once you've done that, we're going to write the codon sequence here. So before we get too far along on this, anyone tell me how to get from a codon to from an anticodon to a codon? Sorry. Okay, backing up a little bit. Anyone know what the four base pairs in DNA are, or RNA? A, T, C, and G. Is that DNA or RNA? DNA, yep. So what about RNA? Yep. Yeah, AU. So you'll have G, C. So this is pretty much how they correspond. So A will become U, T will become A, C will become G, and G will become C. Does that make sense, everybody? So when you're going through these three pair sequences, like for A, for example, the anticodon is CGA. Now it would become GCU. Any questions? Go ahead and take a second and get your first and last name written down. But sorry, this is a relatively important part. On the blank paper, you don't want the letters to be on there. So you just want the anticodon. So in the work area down here, that's where you're figuring out what the DNA codon will be. And then in here, you'll have just the blanks with the DNA codon underneath. So when we rearrange, your partner is going to get the DNA codon, they're going to make it the RNA codon, and then the letter. on here, for that corresponding letter, I would write UGC.
On the codon, do we do it with U's or T's?
So the codon would have T's.
So we're only putting the DNA or the codon on this paper, or we're putting anti-codon name?
We're putting, on this paper, we're putting the codon. I gave you a wrong letter. So that first one wouldn't be a U, it would be a T.
So we're taking this and translating it to that.
No.
Yes.
Okay.
Sorry. So that should really only affect the
Okay.
T versus the
The T
U.
was.
Okay.
Sorry about that.
You're good.
Yes.
So both the codon and anticodon.
Just the codon with T's.
I'm getting confused.
So A is going, if there's an A in the anticodon, they'll become a T in the codon.
So.
We're going
is that-
backwards right now.
But isn't that part of the mRNA?
The codon is DNA.
So like if
Then
you
we're
have
next the
an
anticodon.
So what's
So-
the thing in the ribosome called? Isn't that the anticodon? Yeah, isn't what the tRNA thing?
So right now we're going backwards from the mRNA to the DNA.
Oh, okay, so this.
So then your partner is going to go from DNA to the mRNA and then to the letter.
Oh.
I don't know if this will help, but I like what Joanna did. She circled the letters corresponding to her name or the codons there. That can be kind of helpful.
So after we get our name, what do we do?
So on here, you'll be doing this kind of thing. So if you would be taking those off and these off. And so
You just put. the
your
codon.
partner will only get blank spaces, kind of like you're playing hangman, with the codon underneath. So you won't have any U's on your blank paper. They'll all be T's.
Very long last name.
And if you happen to finish it before everyone else is done, you can kind of like take some time and decorate it. Ideally, these will be kind of like posters that hang up for the remainder of the unit that are personalized. Some long names. So on here, you'll have just the blanks corresponding to each of the letters, and you'll have the codon underneath them.
Wait. I think we're gonna come up and throw that.
Not
Or
quite.
we just switch them with the.
I'll kind of distribute them around just so
Oh,
you
okay.
don't know. The idea is to not know who the other person is.
Ah.
So you guys are going to switch between.
That's not fair, because I was like,
Or so it's-
Yeah, and if you guys are done, you can just like doodle around. I ideally would have colored pencils, but I didn't have those as well.
There's more than movers in
Let's see if we can wrap this up in the next minute or so.
Wait, don't, Scooby-Doo, Minnie Mouse. There we go, Mickey Mouse.
Who all is done, if I may ask? All right. Close. Sweet. Yeah.
Big square back, really big. What is that?
Yeah. No, I like it. All right. If everybody's good, let's go ahead and stop the doodling, decorating, and I'll collect these. I can take these three. All right. Okay.
Oh no, I just did it.
Oh yeah.
So you
Oh, yeah,
turn away.
perfect. Getting me ready. You guys all.
I know.
Okay. So let's go ahead and. That's not yours, was it?
No.
Okay.
Looks like
I think we should be okay. I don't believe either of those are either. And these are 50 fingers. And so the idea now is to work backwards. So we have the codon, transcribe that back to the anticodon, and then to the corresponding letter.
We're about to see.
All right. Pretty. I like it. All right. All right. Almost there.
Here is a cell,
Whoa.
the basic unit of all living tissue.
Sorry about that. We still need more time. Right. So as you're finishing up, you can go ahead and pass your paper back to the person.
Oh, wait, you two have the same one? How cute. I did it.
So the whole idea of this is just kind of get you guys all familiar with the process of going from codon to anticodon, and then eventually you'd be translating that back into DNA. So it can get pretty confusing, and there's a whole bunch of different letters involved. Once you start adding tRNA and amino acids into the equation, it can get really, really confusing. So the whole idea of this was just kind of get you guys familiar with it, get used to that process. So as we go through the rest of the unit, you guys already kind of have a head start on how all of this works. So what we just did was transcription. And so this is just kind of a nice little animation to kind of draw all this together. You guys can see what this actually looks like.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
And that's where we're going to go ahead and stop it for today. Not the most exciting voice, but I think it's a pretty cool animation. Just kind of get a cool picture of things. So for the last thing in this last minute and a half or so, if you don't mind flipping over to the back, and under the take 6, there's an exit slip of sorts. So if you guys can fill that out to the best of your ability. I know there's kind of a lot. Do your best with the time that we have. You guys are finished with those, I can collect them. And I'll actually take your guys' pictures as well. I'll really show us the best photo is. Thank you. I'll take both of them actually. If I had the liberty to do so, these would all be hung up and be nice, beautiful pictures on the walls for the remainder of the lesson. It's just kind of like a fun reminder of the process.